have the best job and the easiest job is I'd like to kick us off and invite the first two interns, Ashley and Abby, to the stage to teach us about what we're really here for.
Hello, everyone. My name is Ashley, and along with Avinash and Abigail, we had the pleasure of working on the GTM Research Reserve project. So I would like to begin by taking a little poll of the audience. Not that I can really see your hands, but please raise your hand if you have ever donated any money to a favorite charity, and keep your hands up. And then raise your hand if you have volunteered at a local charity or agency. And lastly, if that didn't get any of you, you live in Florida, so raise your hand if you have ever saved a tortoise from a busy street. You can put your hands down now, thank you. So in these ways, you've impacted your society in a positive way, and for that, the world is a better place. GTM Research Reserve offers additional ways that you can continue to impact the world through citizen science efforts. Citizen science is a call to action, and it can be as simple as whale watching or reporting a sea turtle nest. The reserve is comprised of 3 core areas. Researchers at the reserve are employed, and they systematically monitor the ecosystem as part of a larger network of 29 national estuarine research reserves across the U.S. Additionally, volunteers support the stewardship program by maintaining research reserve facilities and restoring natural habitats. Educators, students, and the general public can visit the reserve and participate in a variety of educational programs to learn more about their environment. And all 3 of these areas are supported by the Friends of GTM Research Reserve, which is a private nonprofit citizen organization. So our project focused on the education program, which serves over 40 schools and school groups. They provide educational experiences to children ranging in ages from pre-K through college, through 4 Living Lab series and summer camps throughout the year, where they collect both water quality and biodiversity samples and train the students in a research perspective. It's in the education program that the wicked problem was revealed to us. All of this data being collected by students is currently not used for any purpose, scientific or research otherwise, but there is a great need for this research collection. There are certain areas of the reserve where automated instrumentation isn't available to be used, and so manually collecting data samples are the only way that this is done. But the researchers that are imparted with this job do not always have the necessary resources or time available to do so. What if the student data already being collected could be used for scientific research purposes? It's here that we see a wicked problem become a wicked opportunity for a partnership between research and students. But before student research can be deemed usable for scientific purposes, it must first be looked at for precision and accuracy. So we chose to look at student data from the Research Reserve and assess it on both precision and accuracy. And I'd like to invite Avi now to talk to you about how we went about analyzing the data.
We focused our analysis on water quality and compared student data to 2 other data sources. Here you can see the entirety of GTM Research Reserve. It spans from Ponte Vedra Beach south to Palm Coastal, covering 74,000 acres of coastal land. We're going to focus on northern section of GTM Research and Reserve. The purple marker indicates the location where NOAA data sound is located on Tolomato River. The data sound is an instrument that is kept at the depth of 12 feet. The data sound collects a water sample every 15 minutes. The researchers at GTM maintains and quality controls before submitting the data to NOAA. The orange marker indicates the GTM visiting center and facilities. Let's zoom in to that section. You can see the dam. The dam periodically opens and closes, letting the water flow from north to south of Guana River. And the red marker is the area where the researchers and student collects the sample. And this is known as Guana Lake. The black marker indicates the southern side of the dam, where the researchers and students collect the water sample data. And this is also known as Guana River. Due to the similarity of the data collection by the researchers and students, we chose to analyze the data. And you can see here the Pine Island NOAA data zone and GTM visitor centers on two different water bodies, two different locations, and they follow different sampling techniques. Because of that, we took the NOAA data zone data as a baseline comparison to student data and researchers' data. These data sets did not come without challenges. One such challenge is the sheer volume of NOAA data when compared to student and researchers' data. NOAA data has 250,000 entries, where researchers' data has 4,704 entries, and only 323 entries of student data. As Ashley mentioned, we chose to assess the student data based on accuracy and precision. However, data must be precise before it is deemed accurate. So precision refers to how close the data is distributed in the data set and between the groups. A box plot, a standard way of visualizing the data distribution. Here you can see the box plots of 3 data sets on 4 different variables: water temperature, pH, dissolved oxygen, and turbidity. The orange box is interquartile range. The larger the box, the more variable is the data set. And let's look into one of the variables, that is water temperature. You can observe here the researchers' data and student data interquartile range is almost in the same range. And this indicates the similarity of variance between these groups. Additionally, we align these with the NOAA data. And one thing I want you to observe is the outliers. There are more outliers in student and NOAA data, but researchers' data doesn't have any outliers. Researchers quality control their data in the field. When they take the measurement, if there is abnormal reading, they take again. That's how they quality control. And when it comes to NOAA, they have a robust quality control process. They quality control post-collection. That means the raw data is collected and then put through a quality control automatic process, and the researchers flag abnormal and inaccurate data. The flags are left in the data set. Those are the outliers. But the researchers can filter those flags and use the clean quality control data. And student data, because we don't have a similar process or robust process like NOAA or researchers, we recommend to have that processes so that the student data can be more precise and accurate. Now, precision. What we did is, after we matched the data of 3 data sets to date and time, we ran a Levin's test to understand the similarity between the groups. What we found is, out of 4 variables, water temperature, pH, dissolved oxygen, and turbidity, we found that 3 of them, water temperature, dissolved oxygen, and turbidity, had the highest degree of precision in the student data. Ashley now will speak about the analysis that we did on accuracy.
Okay, so accuracy refers in this case to how close the data point is to a desired or correct result. And in order to assess accuracy for student data, we chose to do so visually. Here you can see the distribution of water temperature over time, and this is known as a control chart. It allows us to accurately pinpoint data points and to then compare point to point. So the gray points here indicate the NOAA data. The orange represents student, and the blue researchers. And as you can see, researcher and student data closely follow the trends set by NOAA. However, you will notice a student outlier. If it is removed, the student data once again shows a very normal distribution in terms of NOAA and researcher data. So let's zoom into one particular time period here. And if we zoom in, we can once again see two student outliers. Yet again, if we remove them, the student data shows the same trends as NOAA and researchers. And this is to say that if student data is quality controlled in the same ways that NOAA and researcher data is controlled for, then we can deem student data to be accurate, as we can see here. Let me remind you that the data that Avinash and I have been speaking on and the data that we worked so hard to analyze is mainly collected by middle and high school students acting as citizen scientists. And with this, there come limitations and recommendations for the education program. So the students currently use very similar instrumentation to the researchers, and they collect the same water quality samples. Otherwise, a complete comparison of the data sets would have been next to impossible. However, the students have less availability during summer and winter vacation times, and when students and teachers are scheduling field trips, the teachers may choose different education programs with differing foci, which can impact the sampling times. As well, the student data, when the students go out to collect it, they're trained on site, and they also do not, as we have repeatedly mentioned, have a quality control system currently in place, so this may impact the quality of student data. However, with this being said, we have recommendations for the education program, which include to proactively train students prior to visiting the reserve and to create routine schedules for data collection, as well as quality controlling the data post-collection, and to have an ongoing and open conversation with researchers at the reserve in order to communicate about collection times, methodologies, and results. And so it is our hope that with the improvements of the education program and the student data collection, that this data can show larger trends in the environment and thus be used as a baseline for scientific research. And these improvements should inform citizen science efforts at the reserve, across the nation at national estuarine research reserves, and globally, so that scientific research becomes a more robust process with impacts made by people like you and me. Thank you.